Going to talk about essential plant nutrients today. Let me share my screen here. So when we talk about these essential nutrients, the question is, what's essential? A chemical element that's on the periodic table has to be necessary for plants to complete their life cycle, or it has to be an essential part of a structure of the plant. And also that there can't be any substitute for that. So that's essential. We also have beneficial nutrients, and they're shown to be useful in a plant process, although they're not necessarily essential. I'll give you some examples of that here. If we look at textbooks, there's anywhere from 16 to 18 essential elements, depending on who you ask. According to this, there's 17, but nickel and cobalt are kind of the latest additions. In my opinion, cobalt is essential. It's especially essential for legumes, no question. Probably beneficial at least for others. And we're just going to consider that as essential. I'm going to have you memorize that as being essential. A couple of examples of some beneficials: silica, selenium. However, I've grown lots and lots of hydroponic plants over the many years, and I don't put any silica or selenium in the solutions. Now, they probably get a little bit from the seed, but it's pretty minimal. So we grow beautiful plants hydroponically, and it just kind of emphasizes to me that, yeah, maybe we have research that shows these are beneficials, but not really essential. Sodium's an interesting one because it's so common in the soil environment, irrigation waters. It is used in various plant functions. It is an important consideration when we talk about soil physical properties, chemical properties. But the thing is there are substitutes for what it does in the plants, and therefore it's not essential. But because it's so commonly found in soils and plants and irrigation waters, we generally find it on reports, soil analyses, water analyses, plant tissue tests. So it is an important element that we talk about, but it's beneficial. Now, I have this mnemonic that I made up. Now, originally, the Hopkins Cafe was made up by somebody else. It wasn't me, even though my name's on it. My last name's Hopkins, obviously. But I'm not the one who coined this. I've modified it, though, to account for some of these later additions. So my version of this is C. Hopkins Mongolian Cafe, nickel lunch. The mob will be coming cousin. And so if you look at that, the red letters are the nutrients. And so if you're having trouble remembering these nutrients, I think this can be kind of helpful. Let's just kind of walk through it. The first three, CHO, are non-mineral nutrients. The next three, PKN, are primary macros. Sulfur, calcium, magnesium, secondary macros. And then the next group are the micronutrients. So they're listed, not only are they listed in the mnemonic, but they're listed in the order of those categories, which those categories are important to know. When we talk about these non-mineral nutrients, we need to realize they primarily come from the water and the air, non-mineral nutrient sources. Carbon and oxygen make up most of plants and other organisms by weight. The vast majority is what they are made of. Again, plants are getting carbon dioxide from the atmosphere, and they use that in photosynthesis, and that's where the carbon comes from. We don't have to add it. It's already there. Every one of us is breathing that out right now to help fertilize plants. Oxygen comes from the air as well, with oxygen gases, and also oxygen that comes from water. Hydrogen actually is the number one atom in any organism in terms of numbers. But because it's so small in relation to calcium or carbon or oxygen or whatever, it just doesn't make up as much of the weight. But still, like 5 to 8% of the plant by weight is hydrogen. But again, ubiquitous in the environment. It's everywhere. We don't have to fertilize with this because there's plenty of it all over the place. Now, the mineral nutrients, again, no surprise, come from minerals as well as soil organic matter. We talk about these in a couple of different categories. We talk about macronutrients and micronutrients. Macronutrients can be thought of as in terms of concentration percent levels, going from about 0.1% up to about 5%, sometimes higher. Like potato petioles can be 11% potassium, for example. But in general, they're kind of in that, less than 5% range is where their concentrations lie. The micronutrients are we talk about them in parts per million. Now, parts per million is something that maybe is not as intuitive for most of us as percent. Most of us understand what percent is, right? If I have 1 part of 100, that's 1%. Well, if I have if I've got a million somethings and one of them is different than the rest, then it's 1 part per million, whatever that is. So it's same concept, just a different scale. And it's useful to be able to talk about micronutrients in terms of parts per million because the concentrations are so small. So instead of saying 0.0001%, we can give the parts per million. It's a little easier to grasp what the value actually is. So these range from anywhere from about 0.1 to over 1,000 parts per million. Chloride is kind of an exception. It's actually found in fairly large quantities in plants, but nevertheless, it's still a micronutrient and classified as such. We also further divide the macronutrients into primary and secondary. Primary ones is NPK, nitrogen, phosphorus, potassium. And it's not based on how much is in the plant, because frankly, there's usually or commonly more calcium in a plant than there is phosphorus, for example. But it's which ones are most likely to be deficient. Primarily deficient ones are NPK. So those are primary macronutrients. Secondary are sulfur, calcium, magnesium, not as commonly deficient, but still, at fairly high levels in the plant. On a fertilizer bag, we get nitrogen. Now, nitrogen is by far the most commonly deficient nutrient in terrestrial ecosystems. About half of world fertilizer cells are nitrogen for a very good reason, because we don't have enough when we try to grow, especially crop plants. And so it's really the most important one. Phosphorus and potassium kind of make up most of the rest. The micronutrients and the secondary nutrients, there's not as many of those sold, although it's increasing in number. But really, these three make up the bulk of fertilizer cells globally. Now, by law, if you have a fertilizer bag like this one, it has to have these numbers on it. These three numbers, it can also include the other nutrients, but those aren't as regulated as these three are. But it has to have these three in order. First number is nitrogen, percentage. Now this image actually has got some incorrect things on here with the phosphorus and the P2O5. I did that on purpose because there's so commonly this mistake is made. But the reality is that the phosphorus is expressed not as P, but as P2O5. Now, P2O5 is not how plants take it up, and that's not the form the fertilizer is in. That's what we thought it was when fertilizer laws were made, and we're kind of stuck with it. So what's important to know is sometimes we have to go back and forth between these two, because when we're talking fertilizer, we talk P2O5, but when we're talking plants, water, etc., we talk about how the concentration of phosphorus, P. So in order to make that conversion, it's 43.7% phosphorus in P2O5. The rest is oxygen. Similar for potassium, the fertilizer law requires it to be reported as K2O, which is 83% K, and the rest is oxygen. So that's some interesting and good stuff that has to be on a fertilizer bag. Now, the secondary macronutrients, even though historically these have not been very commonly deficient, sulfur is beginning to be more commonly deficient in the environment. Partly because we're cleaning up the air, we have less pollution, less acid rain, less contamination in various products that we put down, like fertilizers. And so and also yields are going up, and plants are growing faster, so demand is higher. So as a result, we're getting more and more sulfur deficiencies. Calcium and magnesium are still pretty rare in terms of being deficient. They're both ubiquitous in the water and in the soil, and thus are very rarely deficient. Now, there are cases where we get deficiencies. You do have some soils, typically sandy, low organic matter soils, where we're not irrigating, high rainfall areas. Those are the ones, low pH. Those are the ones that will be most likely to have deficiencies of these. We also get calcium and sometimes magnesium deficiencies in some other unique situations, like even in the West where we have loads of calcium in the soil and in the water. We still get some calcium problems with, like an apple, we get bitter pit, or sometimes maybe even in potato, just because of some uptake problems. Micronutrients, they're just as essential, even though they're found at much lower concentrations, but they are essential. Plants have to have them to get their job done. Now, if I'm trying to determine, kind of do my troubleshooting, let's say I'm trying to decide if I have deficiencies or not. One of the first things we would look at is a visual assessment. If I see a plant that doesn't look healthy, one of the questions is, well, is it nutrient deficient, or is it water or something else? But it's good to be able to kind of look at a plant and to say, is it deficient? Now, the problem is this is really complicated, and there are lots of diagnostic keys that are out there. And the problem is there's so many exceptions. It's, one plant does this and the other plant does that, and it's really kind of difficult to follow it exactly. One thing we can say that's pretty common among most of the nutrients is that when they're deficient, the plants are usually stunted. Now, there's some exceptions, like for example, calcium tends to affect the reproductive tissues more than it does the vegetative tissues. So again, there's some exceptions, but by and large, if I have a nutrient deficiency, I typically have shorter plants, shortened internodes, smaller leaves, those kinds of things. Now, just to kind of simplify things, though, we just can't teach everything there is to know about nutrient deficiency symptoms because it's so specific for so many nutrients and so many plants. But the good news is that you can kind of simplify it. The vast majority of what I see visually is really these 5 nutrients, and we'll talk about these. Before we do that, though, it's good to understand this. There's different mobility in the plant for these nutrients. So the primary macronutrients plus magnesium are mobile in the plant. In other words, if that plant recognizes physiologically that it's deficient in any one of those, it'll actually cannibalize older leaves, bring the nutrients from them into newer leaves. And so those older leaves tend to show the deficiency symptoms first. For most of the other nutrients, in most situations, again, it's not a clear cut all the time, but quite often it is that we get these other nutrients show up in the newer leaves because they're not mobile in the plant. So that's a really good diagnostic thing that that's going to be helpful for us as we look at these. So let's just start. Let's look at phosphorus. Now, I picked phosphorus first because it's kind of the weirdo. It's usually not chlorotic. Now, chlorotic means lack of chlorophyll. Chlorophyll is what makes leaves green. And so when a plant lacks chlorophyll or has a reduced chlorophyll content, it tends to be chlorotic. It looks less green, yellow, even white sometimes. Phosphorus doesn't really do that, though, usually. Now, there's exceptions to that, but usually it doesn't show up as chlorosis. We always hear about purpling, and the problem is that corn is the #1 grown plant in the United States, and it is the most studied plant. And guess what? It turns purple. And so the textbooks always talk about purpling. And there are definitely plants that turn purple when they're phosphorus deficient, including, you can see that St. Augustine grass there in the middle picture. That's phosphorus deficiency. But a lot of times it's not purple. A lot of times we get dark green happens. You can see that here in the potatoes. The plant on the right is phosphorus deficient, so it's just a little bit darker green. Now, honestly, if you saw that in the field and didn't have this comparison, you probably wouldn't even notice. You can also notice, though, what I said before is, I've got kind of less plant there. There's less leaves, smaller leaves, and the phosphorus is stunting that plant's growth, even though the visual isn't super apparent. And maybe even a little chlorosis. And notice those bottom leaves on the plant on the right, they're dying. And so that's a clue. Again, mobility. Nitrogen is different. It's one, and all the rest of these are going to show chlorosis. We get nitrogen chlorosis, happens over the whole leaf. It's fairly uniform, tends to appear on the older leaves first, such as this. Again, we're looking at potato here. Potato plant on the right is nitrogen deficient. The one on the left has got plenty of nitrogen. Notice that those older leaves are very chlorotic. Here's another one, or another shot closer up. You can really see that chlorosis on those older leaves and they're starting to die. And here's, a healthy tomato plant on the left and a nitrogen deficient one on the right, again, showing chlorosis. Now, just a word about toxicity. We're not going to really spend too much time right now talking about toxicity, but we can get too much. Now, this isn't really toxic. It's just one of the things that's really common is we get too much nitrogen on. Really a pretty common problem. So the plants on the right, being held by one of my former students, Beth here, those have too much nitrogen. And you'll notice that I'm getting a lot more top growth. It's more green at the expense of root growth. And so that's interesting. So sometimes that dark green color, that really rich green, may actually not be a healthy thing for those plants because we want to have a balance of root and shoot growth. So it's important to be able to manage that as well. All right, let's shift back to deficiencies. Potassium deficiency, also chlorosis, also appearing on the older leaves first. The difference between this and nitrogen is that the chlorosis shows up on the leaf margins. So you'll notice here, for example, in this bottom left photo, that I've kind of, the interior part of the leaf is kind of green, but on the edges of the leaf, the margins of the leaf, it is chlorotic. If you look at this picture over here on the right, these are younger leaves that look pretty healthy. They're on the towards the bottom of the photograph. The larger, older leaves, you can see this chlorosis that's beginning to develop on this leaf, and then on this one in the background, it's really progressed very far on this pumpkin plant. And that's really pretty common. That's potassium is really visually diagnostic, and so is nitrogen, more so than phosphorus, very visually obvious when we have these deficiencies. Shift gears now, sulfur. Contrary to those other three, the NPK, this shows up on the newer leaves because, again, this is not a mobile nutrient. We get chlorosis like nitrogen. It's over the whole leaf oftentimes, but again, different part of the plant. So here on the corn, for example, on the left, you can see these older leaves are a little bit darker green, look kind of healthier, whereas these newer leaves coming out of the whorl are fairly nitrogen deficient. Potato plants, again, over here on the right, you can see this healthy plant on the right, the normal plant, compared to this chlorotic, stunted, sulfur deficient plant on the left. And again, you can kind of see the newer leaves are more affected. That's a little more apparent up here in the leaves as we look across there. The newer leaves here in the middle have worse chlorosis than the older leaves on the left. Iron is the fifth one that's pretty commonly seen. I see a lot, very often, not always, such as the potato here on the right. It's not really, it's kind of more general chlorosis in this particular instance, but usually we get this interveinal chlorosis, such as what you see here with this maple leaf. Now, interveinal chlorosis means the veins are green, and then between the veins is chlorotic, again, occurring on newer leaves. So those are some very visual diagnostics. Again, that's going to be 99% of what you see is going to be those five. So that's very good information to stick in the brain. And then the other stuff, you can kind of work through that extra 1%, do a little digging and detective work. And we can talk about that as we go here. Sometimes we have hidden hunger, or we have situations where it's not one of those five, it's something else, and we need to kind of figure out what's going on with these plants. And so how can I do that? How can I be a detective? A couple of things. I can do plant tissue testing, and along with soil testing. And we're not going to talk about soil testing today, but plant tissue testing is a helpful tool. And we can also add fertilizers to do a little field trial just to kind of verify if I've got certain deficiencies. So let's talk about taking a sample first. Very similar to soil testing. You can look that up and read about how to do soil and plant tissue testing. One of the things that's kind of unique from soil testing is that, or soil sampling, is that you want to make sure you take the correct plant part. I usually tell people to reach out to the laboratory, or this book is extremely helpful. I have a digital copy of this book. It's not terribly expensive. It's got literally thousands of plants in there and what plant part we're interested in. So it depends on which species and what we want. Typically, if I have a really young plant, I'm gonna, I'm gonna take the whole thing. So like for example, I have here this little orchid grown in this little ball that my wife has, and if I wanted to sample this small little plant, I'd probably take the whole plant. Now, if the plant was much bigger, like I've had a whole big 10-foot tall corn stalk, I don't want to take the whole plant. And research shows that the ear leaf, the leaf that's just below the ear of corn, is the one we want. And it varies. Maple trees are going to be different than petunias, and so we need to kind of figure that out. So again, we need to ask those questions. So here's an example of inside that plant analysis handbook. Just here's some different grasses, and these values that are here are kind of what's sort of normal. This is published by a guy who ran a lab, and he's just collected tons and tons of data and has published it and showing you, okay, well, here's what's typical. That can be helpful information. It's not always completely diagnostic for me. Like I might get a value that's below or above these. That doesn't necessarily mean it's deficient or toxic, but if I'm in these ranges, there's a pretty good chance that I've got adequate nutrients. So for example, here's this one here in the upper left. I've got red top grass. This is clippings from new growth during the summer. And for example, 3.3% nitrogen. If you go to this next one, this is a creeping bent grass in B, and you'll notice they actually give us a range. In this particular group of plants, it was between 4.5% and 6% nitrogen. So if I did a plant tissue test and I was 4.55% nitrogen, I'm probably okay. If I'm 4.45, I'm below the range. Does that mean I'm deficient? Maybe not. The further I go below that, the more likely I am to be deficient. If I'm down at 2% nitrogen, for example, pretty good chance I'm deficient. So this is a nice tool, and it kind of takes a little bit of an expert to help interpret this. And so usually laboratories can kind of help with this. Here's an example of a plant tissue sample. This is again turf grass that was taken here at BYU, and you can see the values that we got here. The question is what do these values mean? Are they high? Are they low? Or whatever? We need to have some kind of interpretation. Now, some laboratories will offer that interpretation, such as Servitec. If you ask them to, they will provide some interpretation. Here's another set of data that shows this particular, again, turf grass that was being sampled. So if you kind of look at this is saying, according to Servitec, this is a good lab that I've worked with, and in fact, I used to work for them. I'm saying, hey, the nitrogen, the phosphorus is sufficient, good enough. It says, but it's suggesting potassium is kind of borderline deficient, marginal. Calcium, magnesium are good, sulfur's good, zinc's good, iron is pretty high, manganese is good, copper good, boron good, and again, sodium is good as well, nitrogen sulfurate. So kind of the only thing in this particular set of data would be the potassium. So that would suggest, hey, we probably need to up our potassium in some way. Now, you might want to couple that with soil testing to verify that. And again, this doesn't guarantee it. The other thing you need to realize is that different labs are going to have different interpretation levels. They don't all have some Bible that they go to that says this is the exact range. Every laboratory has the prerogative to set their own limits. So just realize that, and hopefully you work with a good lab who knows what they're doing. Servitec's one of the best. Here's another lab right here at BYU, Environmental Analytical Lab. Again, some more plant tissue on Kentucky bluegrass. This was two different sites, the practice field and the stadium field. And you can see all the different values here, as well as the range. Now, we pulled this range from that plant analysis handbook I showed earlier. And so you'll see here, it's like, okay, the nitrogen is in range. The borons are in range. The calciums are in range. Copper is a little bit on the low side, especially at the practice field. So that's a red flag. That would make me say, okay, maybe we need to look at copper. Here's iron. It's it's a little bit below range. Well, unfortunately, the iron test for tissue isn't great. It's really easy to contaminate it. One little fleck of dust that gets into the sample can contaminate it. And so a lot of times we get false high readings. Now, if I get a really low value, then I might say, well, it is, but we just don't have great correlations with the iron tissue tests like we do some of the others. But anyway, bottom line is it could be deficient in iron. The potassiums are in range. The magnesiums are actually high over range. Manganese are definitely in the above at the stadium, which might even suggest maybe we're a little too high on the manganese at the stadium. At the practice field, it's right in range. Sodiums are in range. Phosphorus here is a little high, which is great. Not too high. It's probably not a big problem, but I certainly don't need more. And then here's sulfur that is above range, and zinc, being the last one, is in range. So that's kind of some helpful information. Again, couple that with soil testing and some other things that we might look at. And you might say, hey, we might at least look at these micronutrients, and maybe we need a little more copper, possibly some iron, and we could play around with that a little bit. So sometimes I see nutrient deficiencies and I'm really not even sure what it is, or I suspect something and I'm not 100% sure, or my client doesn't believe me and I need to somehow verify that. And so we can do some field testing to determine fertilizer need. I do this all the time, where I go out and just do a little trial. Sometimes I take a little fertilizer liquid and I paint it on the leaf and see if I get a green up. I literally will do that. I'll just get a little liquid fertilizer and mix it up and get a little small paintbrush, and I'll just paint it right on a leaf. And if in a couple of days, if that leaf greens up on me, I know that I had a fertilizer deficiency. Sometimes I'll just use like a Miracle Grow complete fertilizer. It's got everything in it, just to say nutrition or not nutrition. If it is nutritional, then I got to figure out which nutrient or which nutrients is the problem. This is a really interesting case study I'm going to show you. This is the church right down by my house. We were being told that they were very frustrated. They were accusing the guys who did the fertilizer of not putting on enough nitrogen. And I said, I don't think it's nitrogen, guys. And so we went down there and did a little trial. Actually, Daniel Peterman, a former student of mine, did this as part of a class project, which was super cool, little project. But you can kind of see where he spread 3 different kinds of fertilizers. The numbers you see there are NDVI. NDVI is Normalized Difference Vegetative Index. It's a nice little tool that we have, and it's not super expensive. And it kind of is a measure of plant health, actually greenness. It sort of quantifies what we're seeing with our eyes. So you can see there that, these greener tissues, like this one here in the bottom right, has got 0.731. The range for NDVI, by the way, goes from 0 to 1, 1 being kind of perfect, which we never hit, and 0 means, dead, bare soil. But you'll see, 0.731 is actually pretty good, nice and green. And then compare that to out here in the control, it is 0.484. That's some terrible numbers, really low. So what's interesting though, is that when I do this little trial, I've got 3 fertilizers. I'm putting on ammonium sulfate. I get a nice green up. So that's getting 2 nutrients, nitrogen and sulfur. Nitrogen from the ammonium, and then sulfur coming from the sulfate. The urea is only nitrogen, and it didn't really green up that much. The ironite, now, even though it's called ironite and everybody buys it for iron, the reality is, frankly, the iron in it isn't very effective. That's a discussion for another day, because when you apply iron sulfate to alkaline soils, which we have here in Utah in the West, it just turns into a rock immediately when it goes to the ground. But the other thing that's in ironite, sulfur. It's got sulfur in it. So I'm looking at this and I'm like, the 2 treatments that had sulfur in them are giving me a green up, and nitrogen only isn't. The conclusion is obvious, that we need sulfur on this property. And so that's really interesting. If you also, if you look around, we did, other places on this particular property, and you see similar results. Although, back here on this particular part of the grass, it wasn't as bad. The NDVI numbers were actually better. And so for whatever reason, we weren't getting as big of a response in this part of the landscape. However, over here in another part of the landscape, pretty similar to the first group, where we're seeing essentially identical results, and it's essentially replicated, verifying our results. So this is a super easy little trial to go out there and do. Didn't cost hardly any money, and you can visually see it. You can be more fancy and use a little NDVI scanner and be able to do the readings. Again, sulfur deficiency. We see these signs. We get these pale green leaves, some dead spots. The veins are lighter in color than the rest of the leaf, actually. And that's kind of one of the traits of Kentucky bluegrass. So another tool to go along with visual and plant tissue testing and soil testing is doing little fertilizer trials. All right, well, that's all I got for today. Growing plants is awesome. I like to see nice, healthy little plants like my little orchid here. We need to understand that these guys need nutrition. It's important, and we need to not just guess, but we're better off when we measure. And so don't guess, test. Thank you.